So now we're on to topic 9, which is everything about cytoskeleton. So reviewing as cells have blueprint, boundary information, interior boundary from interior exterior ability to harness material energy from the environment. They are one of the simplest self-replicating entity that can exist, and they're individual units of life, smallest individual units of life. So like bones that we have, like skeletons, cells also have cytoskeletons. And there is 3 types of cytoskeletons. These are all proteins, so they are all proteins. There's 3 types right here, but they're all proteins, and there's 2 branches. All eukaryotes have, and only animal cells have. So we'll talk about the first type that all eukaryotes have, and that's called one of them is called microtubule. One of the most major, and basically all the eukaryotes have, including animals and non-animal stuff. It's these hollow tubes that looks like this. They're center hollowed tubes called tubulin dimer that form these tubes. Tubulin dimers are also proteins, alpha tubulin protein and beta tubulin protein. These are little monomers, I think. Yeah, monomers that make dimers like this, forming 2 dimers and making these tubes. And these tubes are coming out of centrosome. It's a center right here, dot. Centrosomes are the organizer of the microtubules, which is these long legs, just not legs, arms sticking out in the cell, in the matrix, not the matrix, in the interior of the cell. And they're used for cell division to pull apart chromosomes. They're also there to form spindles originated from the centrosome, and spindle basically a rollet of lots of filaments like this, located near the nucleus, and they are center organizing these microtubules, copying cell division organelles and chromosome moved apart. They are radiate. Yeah, I already said that. So they provide structure. These guys will make the structure work, and they're also for the shape and support. So mainly just skeleton shape and support for the cell. And they're compression resistant. So the reason why cells are somewhat rigid or somewhat compression resistant is because of these tubules, microtubules. They form centrioles in the cell. They form centrioles in cell division. They are the ones that make that form centrioles. I'm still not really sure what centrioles are for. So they They form cilia and flagella for cell movement. So cells that are able to move, that have flagella or these cilia on the membranes and stuff, they are made out of microtubules. Let's zoom out. And there are highways for vesicular transport. That's also very important. One of their major roles, other than shape and structure support, there are vesicle transport highways. So what is oh yeah, I just talked about this one. Flagella, long and few, undulating motion that in the same direction, stuff like spermatozoids. Cilia is very short. There's many alternating powers. So recovery stroke and power stroke. Power stroke is going that way. Recovery stroke going that way. Just any way to move stuff, to make the cell move or individuals move. Now talking about vesicular transport, there is the, like I said, alpha and beta tubulin. So there's positive and negative sides. Positive sides always tubulins adding, and negative sides are always shrinking. And because there's like a shrink and the negative, this is called dynamic instability, and they're able to shrink or increase, and they're also able to move because of that. I think that's why. And there are ATP used to cause this confrontational change needed for walking for these kinesyns. So kinesyns or dyneins. Kinesyne and dyneins, they all look like this. They're like walking stuff. They're super cute. They are kinesyns walking toward plus, and dyneins walking toward the minus, which is the shrinking side. Plus is the growing side. And surprisingly, kinesyns are beta tubulin subunits exposed, so it's towards the beta side, because there's like a beta and the alpha, right? And dynein is the tubulin subunit of alpha tubulin sides. It's just the side. It's just which side is what. And yeah, so the dynein the negative side is the shrinking side. And it's all about direction. So kinesyne will walk positive way, and dynein will walk to the negative way. It's just really the direction of their going. And if there is too much of this growth at the growth are rapid at the plus Yeah, so this is two completely different stuff. Growths are rapid at a plus side, so they are increasing towards the plus side. But it is a catastrophe if it switches from plus side to a negative side. So this is two completely different stuff. Nothing is equal here. Yeah, so why am I able to erase the apple? So if it is able to grow a lot toward the positive side, then there's no problem. But there is problem if they start growing, if they start shrinking a lot on the negative side. So instead of growing, if they start shrinking, that's a catastrophe. Not really sure why it's a catastrophe, but it's a catastrophe. Now moving on to microfilaments. Microfilaments are smaller. They're just smaller stuff. Not stuff. Smaller filaments, not unlike microtubules, which are a lot bigger, and there's only one direction. And these are made out of not tubulins, but actins. Yeah, so these are tubulin monomers. Now these are actin monomers, and they are double helix like that instead of hollow tube. Not a tubule anymore. It's now a helix, double helix, made out of actin monomer proteins. Actins are also proteins, and these provide mechanical strengths, which is a muscle contraction, we'll see later, interact with other protein, adjust consistency of the cytosol, and that we can see from here. Oh, oops. So this is the cell, for example, and this is the microvilli. These are little undulations of skin. And inside them, there's a lot of microfilaments. Microfilaments are in these undulating microvillis. And they interact with proteins because there's a lot going on within them, and proteins always interacted with lots of things, and proteins will interact with microfilaments as well, and they will adjust the consistency of cytosol. So like shrinking them or elastifying them, I think, is going to make the consistency of cytosol change. Interact with thick filaments such as myosin. So filaments are like these leg guys. Leg guys are also called filaments. They interact with those for motor myosin motor proteins for muscle contraction. For example, here, these are actins, and this is a myosin. And by moving these actins together towards each other or letting go, it's how the muscle contraction works. I'm not sure which one's muscle here, though. Okay, anyways, these are extensions of plasma membrane, these microvilli, and inside there's microfilament, like I said, and their function is to increase the surface area. So microvilli is for increasing surface area to facilitate absorption of extracellular material. So extracellular is everything outside the cell. So these guys are on top of the surface of the cell, and everything outside of it is the exterior of the cell. It's outside. And they're attached to cytoskeleton, which is basically everything we're talking about right now. So these microfilaments are also allowed to able to make cells move, like I don't know, worms, and they are doing that by the extension of microfilaments. These microfilaments extend it, and will make laminipodia or laminipodium, which is just walking, stretching, walking lamellipodium cells right here. I'm not sure what type of word this is, but just lamellipodium just means walking of these cells due to microfilament stretching and compressing. They are also able to do vascular support. So vascular support was done over here by the coenzymes and dyneins, but they also do some. Microfilaments also do some. I should say they do totally do, actually. Motor proteins are associated with microfilaments, such as myosin is associated. Well, instead of bringing... Oh, that's interesting. Yeah, so myosins are able to transport vesicles, but also contract the muscles, contract microfilaments. So microtubules and microfilaments, they're both tracks for moving vesicles throughout the cell. So there are other filaments that are going to walk on top of it, and the vesicles are going to be transported, and energy comes from the stored ATP. So the energy required, who requires those energy? So moving on, intermediate filaments. Intermediate filaments are the ones only in animal cells, not all eukaryotic cells. And these are fiber proteins. They're strong fiber composed of intermediate filament proteins. Intermediate filament protein. So these are intermediate filaments. These are fibers, strong fibers composed of intermediate filament proteins. And these are fibrous subunits that make up these intermediate filament proteins. Fibers are made out of proteins. Some fibers are made out of proteins. Yeah, proteins are able to even make proteins, fibers, such as collagen, keratin, made out of these fibers that are made out of proteins. Yeah, anyway, they are mid-sized, so they're sized between the microtubules and microfilaments. They have different sizes. And the tension bearings, what is tension bearing? I'm not sure what tension bearing is. They're able to do tension bearing. So it's for integrity, cell integrity, and resist mechanical stress. Strengths. They're able to provide strengths. Yeah, so they're just able to provide some strengths. Functions of these intermediate filaments: strengthens the cell, like I said. Okay, the tension bearing means strengthen the cells. They strengthen the cells, maintain the shape, structure of nucleus. Remember that structure of nucleus is done by these intermediate filaments. They are holding the structure of nucleus, stabilize positions of organelles, stabilize cell positions. So organelles and cells themselves. Intermediate filaments outside. So I think intermediate filaments are also in the cellular matrix. No. They're not in the cellular matrix. They're not outside, but they are able to be anchored by anchoring proteins within the cell, but with other cells. So different cells can be binded, like anchored, like disconnected, from intermediate filament to another intermediate filament, anchored by these proteins. And the tension bearings, like the microfilaments, are what keeps this really strong structured. So structure of nucleus, stabilize position of organelles, stabilize cell position, cell to cell. So position inside the cell, position outside, so within cells, within their cell, and relative to each other. Yeah, so stabilize cells relative to each other and relative to extracellular space. Okay. That's something I'll keep in mind. So it is, I think it is in extracellular space. Okay. So now we're moving on to the extracellular matrix, which is basically the outside of the cell. So outside of cell, I should say, like the very skinny portion outside of the cell, like a skinny membrane, not a membrane, but like there's a bit of skin, not a skin, matrix, enveloping each cell, and yeah, we're gonna look at what that is. So there is molecular framework, structural architecture of the plants and animal cells, which is called the extracellular space, and there are structure supporting supports that provide information cues determining the activity of cells. So basically, there's cells, and there is the exterior of the cell a little bit like this. This is the extracellular matrix, which are going to signal into what is going on into the cell. So these extracellular matrix are going to signal stuff into the cell, telling what's going on outside, which is the cue information for the cell to function. So they're gonna do other stuff. They're gonna do their stuff depending on what these signals are gonna tell them, and these signals can actually modify their activity because depending on their environment and everything. So these matrix are found in most tissues, but they're most commonly found in connective tissues. So extracellular matrix are found a lot in connective tissue and also a lot in, I think, hurt, injured tissues because there's a lot of collagen and elastins. So I know that there's a lot of collagen in injuries because I'm really injured myself. But these collagens are flexible. Oh, sorry, no, elastins are flexible. And collagens and elastins, I think we want to talk about it more later. Anyway, there is a lot of them. These are the fibrous proteins. Fibrous proteins make up these matrix, and there are collagens and elastins. That's all you need to know. Collagens and fibers are embedded in network woven of proteoglycans, which is gel-like substance of this matrix. So there's a network, there's a lot going on, called the proteoglycans, which is the gel, of which there are other collagens and fibers embedded in them. So it's a mess of lots of proteoglycan gels, triple helix, with protein, fibers, and collagens and elastins, and yeah. So these collagen and elastins are secreted by fibroblasts, an active cell of connective tissue, and proteoglycan. Active tissue is produced by the extracellular membrane, producing collagen, elastin, proteoglycan, structural protein, maintain structure framework of the tissue. So fibroblasts is where fibers are blasted out. Not really sure what fibroblast is. No, I don't know if I even need to know it. Now, glycopeptides, glycoprotein is something we talked about for a while already, but so let's talk about actually the extracellular matrix and the cell. Cells are attached to this extracellular membrane, like I said, and there are stuff that are sticking out of the cell, like glycoproteins, which is among this matrix, and these are bound to cell surface, like here we can see right here there is matrix outside. This is a bilayer phospholipid membrane, and this is the inside, and there are proteins called integrins, and there is the fibronectin, which is a type of glycoprotein. So glycoproteins are the ones sticking out, or I think I should say within the membrane, and the sugar part is sticking out, and that's a glycoprotein that was made by the endoplasmic reticulum, which was secreted out thanks to the vesicle compositions of Golgi, and all that. So glycoproteins are here again. They are able to attach to proteoglycan complex, which is the gel-like matrix complex, attached to integrins, proteins that are within the membrane, attached to inside here. These are microfilaments. The alpha tubulin that we talked about over here, right? Those are here. Now, they hold to stabilize. I don't know what is holding what to stabilize. what is holding what to stabilize here? Yeah, so I think it's really these guys. To stabilize all of these the structures so that basically everything's organized. And the integrins are receptor proteins, meaning any signals that are transmitted to here, to just go into the membrane, into the cell, and delivered to the DNA, into the nucleus to change if they ever need to change their protein sequences, their function, etc. Yeah, so integrins bound to microfilaments. So fibronectin, not pectin, their function is to bind to collagens, function in adhesion, regulates cell matrix and matrix-matrix interaction. Cell matrix, so cell and the matrix, as well as matrix amongst themselves, are being regulated by the fibronectins, which are these guys here. So these guys just have, they bind to collagens as well. They bind to lots of things. Bind to collagen, bind to proteoglycan, gel-like substance, and everything to do signals. Integrins are built into the plasma, so they're built-in receptor proteins in the membranes. They transmit signal between the extracellular membrane and the cytoskeleton. So cytoskeleton is, for example, these guys, to transport information and integrate changes inside and outside of the cell. By communicating with the cell through these integrins, like I said, ACM regulates cell function, structure, behavior, to receive signal and transmit it to the nucleus via microfilaments. So microfilaments where these signals are walking on highway to go to their nucleus. Signaling will change the behavior of the protein. So the proteins that are secreted, that are made, sequences of the proteins are going to be changed by these signals. These signals basically come from matrix into the fibronectin, into the integrins protein, and reach there, and proteins are synthesized and triggering these protein synthesis. So that's it.